Hello everybody. Welcome to our program, The Wonderful World of Drug Design. Today we will talking about SR, QSR and pharmacophore identification. Hi guys, my name is Mr. Benzodiazepine and I will be your host. Today's topic is very interesting, but probably you are asking what is SR, QSR and pharmacophore. Well, let's see what think our friends in the College of Chemistry.
Hi Sarah. What's up?
Hi Henry. Nothing special. How are you?
I'm fine, but we have many homework in the last days.
You're right. I feel that I don't know what is happening in the world. And lately I've been listening too much about pharmacophore and QSR and SR.
Yes. Everybody is talking about that.
What do you think that means?
Ooh, you don't know.
No, I don't.
We don't know.
Maybe it's a new rock band. Or it is a new dance style.
Oppan Gangnam Style.
Not really. I will explain to you what this concept means. First, what is a pharmacophore? Pharmacophore is the set of chemical groups, bonded together or not, which have in common the active molecules of the same receptor and are essential for the recognition by the same. The pharmacophore summarizes the important binding groups which are required for activity and their relative positions in the space with respect with each other. It is not necessary to show the specific skeleton connecting the important groups. Indeed, there are benefits in not doing so, since it is easier to compare the three pharmacophores from different structural classes of compound to see if they share a common pharmacophore. Three pharmacophores can be defined using molecular modeling, which allows the definition of dummy bonds, which is usually named as centroid. In order to identify the three pharmacophore, it is necessary to know the active conformation. Normally, this conformation is recognized and bound by the binding site. There are various ways to find the active conformation. Rigid analogs of a flexible compound could be synthesized and tested to see whether activity is retained. Alternatively, it may be possible to crystallize the target with the compound bound to the binding site. X-ray crystallography could then be used to identify the structure of the complex as well as the active conformation of the bound ligand. Finally, progress has been made in using NMR spectroscopy to solve the active conformation of isotopically labeled molecules bound to their binding sites. It is not uncommon to find compounds that have the correct pharmacophore, but nevertheless show disappointing activity and poor binding. It is important to realize that the overall skeleton of the molecule is involved in interactions with the binding site through van der Waals and hydrophobic interactions. The strength of these interactions can some be crucially in whether a drug binds effectively or not. Well, after this little definition of pharmacophore, now I will talk to you about SR, that is, the abbreviation of the relationship between the structure of the molecule and its biological activity. It could help us determining the chemical groups responsible for evoking a target. Biological effect. Medicinal chemists usually insert different chemical groups to modify effect and potency. The basic assumption for all molecule-based hypotheses is that similar molecules have similar activities. This principle is the base of SR. The underlying problem is therefore how to define a small difference on a molecular level, since each kind of activity, reactionability, biotransformation ability, solubility, target activity, and so on, might depend on another difference. SR widely used in the systematic design and refinement of pharmaceutical agents, and in the identification of structural alerts of toxicity and mutagenicity. The aim here is to discover which parts of the molecule are important to biological activity and which are not. X-ray, crystallography, aforementioned studies with molecular modeling software to identify important binding interactions. However, this may not be possible, either because the target structure can't be crystallized or because the target structure has not been identified. Revert to the traditional method of synthesizing a selected number of compounds that vary slightly from original structure, then studying what effect it has on the biological activity. Together with the SRR exist the QSRR methods. QSRR methods are trying to establish empirical models of behavior on families of biologically active compounds in order to obtain the most effective optimal activity. This is done by correlating quantitative structural characteristics with biological activity. This method relies on the use of series of compounds derived from a common structure, in which the alterations are observed in their biological activity produced according to the presence of different substituents. From these data, it is possible to calculate novel additives for each substituent, and it is possible to design the best combination of substituents to achieve optimal activity. Both qualitative and quantitative modeling methods relating chemical structure to biological activity and are an important step in the design of a new drug. In our process, the last step is when we are identified the important bonding groups and pharmacophores, so it is possible to synthesize analogs that contain the same pharmacophores. An example of structure-activity relationship are the antifungal agents. The mechanism of action of drugs that inhibit fungal growth is related to its chemical structure. The great similarity between mammalian and fungal cells becomes a problem when designing the molecule antifungal. Antifungals are very used to mucosal infections and other affections. These antifungal agents acting in certain parts of the fungus. In the cell membrane, antifungals have azoles and polyends, and its cell wall use lipopeptides. Also, the fungal cell nucleus use them to metabolites and miscellaneous agents. In the antifungal structures, the presence of 5 atom cycles in which the nitrogen or sulfur are part of the cycle could be considered a pharmacophore group. In the absence of these atoms, molecules lose their biological activity against fungi. The most important pharmacophores found in antifungal are the benzene rings with halogen substituents, such as chlorine or fluorine, near a metazole or triazole ring, help to increase the biological response of the molecule. Another structure which has been used to design molecules is one containing antifungal orthogonal-planes, spirocheles, and one example of this is the griseofulvin. The structures forming cycles in which the amide group is repeated also confer antifungal activity to the molecule, as in the case of the lipopeptides. Functional groups. Unwanted medications in general are very reactive functional groups. Hydrophobic functional groups. Poorly soluble and toxic functional groups. The design of drugs with biological activity QSRR studies are of great importance, as well as the susceptibility of the union of all guarantees unstoppable development of antifungal agents. Well, all this time we have been talking about the function of pharmacophores, but which are the principal objectives in the design of drugs? It's important to know this because drug design reduce side effects makes easy the to synthesize, improve easy and efficient administration to the patient, and of course, increase pharmacological activity. For all these reasons, this kind of study about the drug design is an essential part in the discovery of new drugs. And I'm sorry to say you that this is the end of show. Thanks for watching. We'll be waiting for you on the next show. Goodbye.